Now that you know you have made of genes, and those genes are chemical factors that are borrowed from your parents, and they borrowed those from your grandparents. So basically, pass on from one generation to the next. All right, and we also know that those chemical factors have different forms, and genes, the different forms of genes we call alleles. Okay, and we don't always inherit the same alleles or different or the same property of the same genes in our family. That is called dominant versus recessive. And now that begs the question, how do I know, or what is the chance of my future kids being a certain variation of the same gene? That brings us to probability. And probability is the likelihood to have a certain outcome. And how do we study that in genetics? We use Punnett squares. Please do not let the word probability frighten you because of the math context to this word, because this is the word we use every single day in our everyday lives, like livelihood. Like, what is the probability that you will do this, or the probability that you will get this, or what is? It's like saying, what is the likelihood? Okay, what is the what are your chances? Okay, so probability is the likelihood or chance that an event will occur. I have a nickel. What is my probability when I flip it? I will have a head or tail. Well, a nickel only have two sides, so 50%. Well, I got head. Okay? Your past outcomes do not affect the future ones. So not because I just had, head, that doesn't mean the next time I reduce my chances of having head if I flip this coin. Okay? So for example, what is the probability if I flip the same coin 3 times in a row of having heads? It's 1/8. Okay, what do I mean by that? Just simple multiplication of fraction. You multiply the tops. Oops, sorry, that's a big one. You multiply the tops, all right, and you multiply all the bottoms. 1 times 1 is 1 times 1 is 1. 2 times 2 is 4 times 2 is 8. So 1/8. So geneticists use Punnett square to predict offspring characteristics, or the different alleles that an offspring will get from a parent. Okay, and that pretty much explains the probability behind each offspring in a family being able to inherit certain genetic characteristics. Every time a family decides to have a child is a whole new ball game. All right, so the chances that they had a blonde hair, blue eyed kid, or a brown hair, black hair kid the last time does not affect the chances of having the same outcome again in the future. Please know your key vocab terms, as they will make your life a whole lot easier. Okay, heterozygous. Look at hetero. Okay, I said last time, hetero means different. When we talk about heterotroph versus autotrophs, these are two different alleles. Okay, so this individual has one tall copy of the high gene, and also have a short copy of the high gene. Okay, heterozygous are also referred to as hybrid. Okay, homozygous. Homo always means same in science. So this is an individual with 2 of the same alleles. So they either have 2 dominant copy of the tall gene or tall allele, or you are 2 copies of short, so you will be short. When you have exactly 2 of the same allele, that is also referred to as a purebred, or is a form of true breeding. Okay. Phenotype. Phenotype is the physical characteristics of a gene. It is the part that makes you see that gene. What do you see? I see tall plant. I see short plant. I see brown skin. I see light skin. I see dark skin. I see brown eye. I see green eye. I see blue eye. I see curly hair. I see straight hair. All those characteristics are all physical characteristics. They are things that you can actually physically describe. All right, if it helps you, physical starts with a P, phenotype starts with a P. Okay, genotypes. Genotypes are the genetic makeup of a gene. You don't see a person's gene. You don't look at yourself, oh man, I see your brown eye gene. No, you don't see that. You can't see a person's height gene. Okay? We don't see that. We only see the way we represent it is we just represent them in a letter combination. Genotype, genetic starts with a G, genotype starts with a G, if that helps you at all. So probabilities work better when we actually have more trials. And generally, it's a general rule of thumb is to do something in science maybe 3 times, at least 3 times, 3 times, because 2 out of 3 times, if you have the same outcomes, then maybe it's worth further investigation, okay, or not. All right. So we will have a penny lab, and basically it's good old penny lab, and see how many times you can create the same outcomes. So basically, probability now leads us into Punnett square. Punnett square. Punnett squares are actually named. Punnett is the name of the guy who came up with this whole idea of how to determine genetic probability. So when you look at this parent generation, they are both hybrids. Okay. Also, they are both also known as what? Also, what's another H word? Heterozygous. And I can't fit it on the screen. All right. The way these are done is that you have one parent. The parents are separated to the top, okay, across the top, and along the side. So when you read a book, you read from left to right, top to bottom. Use that same mentality. Take everything from the left and carry it straight across. And take everything from the top and bring them straight down. Okay? And you will have exactly the same outcome as on here. Oops, sorry. That's supposed to be blue. There you go. Okay? So, big T here, big T here, big T here, little t here, little t here, big T here, little t here, little t here. All right. So you always write the dominant letter first. That's why this T actually came second. Okay. All right. Now here's more practice problem. A green pea plant is being crossed with a green pea plant. Whoa. If I just give you a statement like that, that makes no sense whatsoever. All right. So, well, they're green, so why should I care? Predict the genetic and phenotypic outcome of the offspring. Well, that's why we have the little ones in the parentheses. I will always give you that, or I'll give you a descriptive word. So, big G, big G. This parent is a homozygous. Okay, homozygous for grain. Also purebred. This parent is a heterozygous. Also a hybrid. All right. Right in with your finger on the screen. It's a little harder than you know it. So let's take the parent from the top, or this parent, and move them here. Move the parent here. So we have big G, big G. And now let's take the other parent, the heterozygous parent, big G, little g. Okay? So I will go ahead and write in all mine left to right. So I'll go big G, big G. Alright? Remember, I would have to leave room for the big G because little letters always come second. I write my little g, and I write my little g. Alright? I go back and change color and say, now let me bring from my top to bottom. So big G, big G, big G, big G. Okay? So now What are all the different genetic possibilities of this cross? What you look here, we have right here, big G, big G. Okay? How many of that do we have? 2. So that's 50%. Oops. Percent. There you go. We also have um, big G, little g right here. We have 2, they also have big G, little g. And so that's also 50%. So the outcome of this couple actually making a homozygous dominant offspring is 50%, and also a heterozygous dominant for grain is also 50%. Now, look at the same property again. All right. So now this time, a green pea plant is crossed with a yellow pea plant. Okay. Predict the genotype and phenotypes of the offspring. Well, there are several assumptions in this statement. Okay. We know that because we have a capital G here, so green has to be a dominant trait. Okay, and please don't get lost. Okay, not because yellow is here, that doesn't mean we have a small y. The lowercase letter is also the lowercase letter of the dominant trait. Okay, so the dominant trait here is green, and therefore we use G. Okay, and to represent yellow, we're going to use lower G. So let's go ahead and put our parents into the right places. This parent is down here. We've got big G, we've got little g. All right. This parent is down here, little g, little g. Just for vocab practice, this parent right here is that parent is a heterozygous, also hybrid. This parent right here is a homozygous, also purebred. So when we go through, I will add in all my little g's first, and now I'm going to go back and change color and add in my big G's. Oh, sorry. Oops. This is little g, by the way. Sorry. This is little g, by the way, because it's coming from on top. Okay, that was a good mistake. So, learning moment. All right. Now, what are all the different genetic, characteristics? When you look, and we didn't answer this second part of the question in the last practice, which we're gonna do here now. What are the different gene combinations we have? Oops, sorry. We got big G, little g. Okay, we have little g, which would be these guys. My messy handwriting. Out of 4 outcomes, what is the possibility to have big G, little g, or the green plant? Is 1/2, and this also is what's 1/2. Okay, how do I get 1/2? 2 out of 4. Okay, when you have big G, so what is the likelihood to have a green plant, and what is the likelihood to have a yellow plant? Well, to have green, we have how many with capital G? 2, right? So these 2, so that's 50%. Also 1/2. Okay, what is the likelihood to have yellow plants? We have these guys right here. All right, so we have also 50%. Also that is also 1/2. Simple. And now there are times I will give you problems like this, okay, where you actually have to write the genotype combinations based on your understanding of the vocab terminology. Okay, so now we have heterozygous red flower is crossed with a white flower. Whoa, how do I know which one is dominant? What color is dominant? All right, think. So when you have a heterozygous, you have one big letter and one small letter. And because you have a big letter in that combination, therefore that has to be the dominant trait. So how would we write a dominant, a dominant, a heterozygous for red? So we have a big R and a little r, okay? That's red, okay? And white flower. Well, if red is dominant, then therefore white has to be recessive, okay? So having to represent white flower as a recessive trait, we use lower r. All right? So that's equal white. All right, now look at it this way. Time to cross the parents over to where they need to be. This one comes on top, so we have big R, little r. This one on the side, this one moves on the side here, we have little r, little r. So let's read from left to right. Little r, little r, little r, little r. This kind of looks like a V, sorry. Now let's read from top to bottom. All right, we got big R, little r, little r. Whoa, great. So now tell me, what are the genotypes? So I'm actually going to introduce something that I want you to be doing from this point on, is write things out so it can make sense to you. So genotypes, underline that. Somewhere else write phenotypes, and kind of like underline that. All right, so let's look at the genotypes, the letter combination. What are the different letter combinations do we see in those 4 boxes? Okay, we have big R, little r, big R, little r. So I've got big R, little r. Okay, we also have little r, little r. Okay, let's take one step back. Each of these boxes represent 25% probability. Okay, so if you got a little taken back when I said 50%, that's what I mean by that. Each box is 25%. So now, how many boxes of little r, big R, little r do we have? Okay, we have 2 of those, so we have 2 out of 4, which is what? 1/2. Okay, which is 50%. And the other one we have 2 out of 4 also, these 2 right here, okay, which is also 1/2. Okay, what are our phenotypes now that we know what are the different gene outcomes? Well, if we have big R, little r, that means what? Red. If we have little r, that means what? White. Okay, and therefore we have a 50% chance of having a flower with red, 50% chance of a plant giving red flower, and a 50% chance of a plant giving white flower. Okay.